Hello. So today we'll talk in this lecture on two major topics. Since we discussed in the last lecture how DNA is synthesized, so in this lecture we'll discuss the transcription of RNA, which actually how RNA is synthesized, and we'll describe the structure of different RNA and how the transcription of eukaryotic genes and the difference between the eukaryotic and prokaryotic RNA. As well, we will have a general difference between the eukaryotic and the prokaryotic transcription. As well, the second topic in this lecture will be the RNA processing, and we will describe how the native or the premature RNA, messenger RNA, are processed in the nucleus before migrating to the cytoplasm in order to do the translation. We'll start with the first topic. So generally, there are a central dogma that says that it's very understood that DNA is transcribed into RNA, which is translated into protein. So actually, DNA contains a gene, a set of genes, and actually the genes are the healthy unit of the DNA. And it contains all the information actually that specify the synthesis of functional RNA to build the protein. So the sequence of the amino acid which build the protein actually exists and they encoded by the transcription and translation of the genes. And you have a 4-base language in the DNA, which is AGCT, that is transcribed into another 4-base language, which is AGCU, with a difference with the U only. And then this language of the messenger of the RNAs are transcribed into a 20 amino acid language of proteins. The primary structure of RNA is very similar to that of the DNA with two exceptions. The sugar of the DNA is a ribose, and you can see from the figure here, let's have a pointer. You can see the ribose differ that these at the 2' position, you have an hydroxyl group here, which is lacking in the deoxyribose sugar of the DNA. As well, in the DNA, you have a thymine T, which is replaced by a U in RNA. So what makes RNA or the ribose or the sugar in the RNA have this hydroxyl group at the 2'? The first thing we need to know that the presence of T rather than U in DNA is important for the long-term stability of DNA. As well, the OH, which exists in the ribose sugar of the RNA, makes the RNA more chemically labile, so it easily can be digested. It's less stable than the DNA. So this OH will provide a chemically reactive group that takes part in RNA-mediated catalysis, so that the RNA can be cleaved by mononucleotide by alkaline solution. And as you know, usually RNA, when it finishes its function and got translated to the desired protein, it needs to be cleaved into a nucleotide to be recycled again for new messenger RNA. So like DNA, RNA is a long polypeptide, and it can be double-stranded or single-stranded. The general belief it is a single-stranded, but sometimes the RNA can fold on each other to form a double-stranded structure. It can be linear, it can be circular. So there are 3 general types of RNA: the messenger RNA, the 3 famous ones, the transfer RNA, the ribosomal RNA. RNA can form a hybrid helix. Sometimes there is some situation inside the cell that RNA can bind with a DNA, as we will see later. RNA, and RNA-DNA double helix have a conformation of A form. If you remember from our first lecture that the conformation of the DNA normally in our cell are in the B form. Yeah. Some RNA molecule have its own built-in catalytic capacity, and these RNA are called ribozymes. They can can catalyze self-splicing during the formation of the majority of functional RNAs. So some RNAs need this self-splicing in order to be functional. In addition, of course, to messenger RNA, you have ribosomal RNA, you have transfer RNA, two other major RNAs, genes. There are other 7 of additional genes that can be transcribed to non-coding, non-protein coding RNAs. So they are usually transcribed and they are not translated, like the small nuclear RNAs, like the small nucleolar RNAs, like the large family of microRNAs. Up till now, they discovered around 2,000 in human of short microRNAs. So these 3 types, either the small nuclear RNAs, the small nucleolar RNAs, the microRNAs, are transcribed from genes on the DNA and they are not translated.
Yeah, they have no function?
No, they have a lot of function. They have a lot of functions. Okay, and recently, in the last 10 years, they found that a lot of microRNA are correlated with cancer. Some RNAs increase, microRNAs increase in presence of certain type of cancer, and other are decreased. Even they are regulate the expression of the translation of messenger RNAs, the microRNAs. Unlike DNA, of course, RNA is single-stranded and have a variety of conformation. For the conformation, of course, you know, the primary structure of RNA should be single-stranded, but sometimes you have like a hairpin structure of RNA. So the RNA fold around itself in a region of 5 to 10 ribonucleotides, and you can see this hairpin structure, and it can form a double-strand structure, a double helix, which is RNA helix, double strand. Other can form a stem loop secondary structure. The only difference between the hairpin and stem loop, that the region of the loop here is 5 to 10 nucleotide, ribonucleotide, but here it can go from 11 to 100 ribonucleotide to form the loop and the stem. So that's why it's called stem loop. And in this region, you can see it's a double helix region of RNA double helix region. A tertiary structure may arise in which these are a combination of 2 stem and loops. You can see stem 1 here, and it's loop 1, and you can see stem 2 here, and this loop 2. And actually, although it appears like it's a knot, but actually it's a pseudo knot, because if a messenger RNA undergo this tertiary structure and formed a pseudo knot, so it had to be untied or relaxed and converted to a single strand structure in order to be efficiently translated. شباب، في حد عنده أي سؤال في الجزء اللي فات لغاية دلوقتي؟
طب التيرشري ستراكشر شو مشكلتها ده؟
مش مشكلة ولا حاجة. It's a conformation of the RNA. And of course
يعني عملها translation.
Of course كل الاستراكشر دي بتحصل نتيجة للcomplementary bases. اوكي؟ اوكي؟
اوكي. يعني هدول اشكال للار ان اي صح؟
اشكال الار ان اي once it's not in action. لو الار ان اي
اوكي.
لو افترضنا ان الار ان اي ده هيحصل له ترانزليشن، لازم يتفك، لازم تتفك العقده دي. فهمت؟
بتصير لينير.
لازم تبقى لينير. اوكي. نكمل. So let's see here how the transcription goes on. As we knew from the last lecture that DNA synthesis goes from 5' to 3', as well transcription goes from 5' to 3' by RNA polymerase. So here you can see the template of the DNA goes from 5' down up till 3'. So to do transcription of RNA, it has to go from 5' to 3'. So here, start the nucleotide is added, of course complementary to the template DNA after it have been melted. So the gene region is melted, and RNA polymerase start to add RNTP, which is ribonucleotide. So you can see it add the base, and the strand growth starts from 5' of the new strand, and the bases are complementary and goes on until we usually have a 3' end. So if we look closely to an incoming, ribonucleotide, so it have a base, it have a sugar, it have 3 phosphates, alpha, beta, and gamma. So this incoming ribonucleotide have to be added by the RNA polymerase to that position. So you can see that the complementary bases bind by hydrogen bonds, and then this need to be attached to the phosphate sugar backbone. So you can see there is a bond will be formed between the OH on the 3' end and the alpha phosphate. This polymerization will yield a high energy bond. This polymerization will relieve the beta and the gamma phosphate, and the breakage of this bond, this high energy bond between the alpha and the beta, is replaced by a lower energy bond, which is between the alpha and OH. This will relieve a pyrophosphate, which actually 2 phosphate groups, by the help of the enzyme pyrophosphatase, which by the help of the pyrophosphatase, it will degrade the pyrophosphate into single phosphates. So again, the incoming ribonucleotide in the growing chain of the RNA will replace the high energy bond between the alpha and beta phosphate, will replace it by a lower energy bond between the OH and the 3' and the phosphate of the alpha. And the pyrophosphate released, which is actually 2 phosphate groups, by the action of the pyrophosphatase, it will be catalyzed into 2 single phosphates. حد عنده سؤال يا شباب؟
لا تمام.
Okay. Again here, let's understand some nomenclature here. So normally you have نعم
كثير فهمت اللي قبل.
ما ايه حضرتك؟
ما فهمت اللي قبل، السلايد اللي قبله.
نرجع كده. طب في حد كده مصحصح معايا ومركز كده يقول لي يشرح لي ايه اللي حصل في السلايد دي؟
انا بالله.
اتفضل.
الامر ان اي هو من ال 5' لل 3' اند بيصير باي ار ان اي بوليميريز. هلا النيوكليوتايد بيس هي معموله من 3 فوسفاتس الفا بيتا وجاما مع الشوجر ومع البيس. لما بده يصير في كونكشن بين التو ار ان تي بيز يلي هن النيوكليوتايد بيس، بينكسر البوند بين الالفا فوسفات والبيتا فوسفات، بينكسر وهو هاي انرجي بوند. بعدين بصير في a weaker bond between الالفا فوسفات مع ال 3 الكربون 3 اللي هي على الشجر الثانيه. بعدين في بيجي بايروفوسفاتيز يكسر التو فوسفاتس البيتا والجاما، بيعملون كل وحده لحالها، فوسفات بيتا لحالها وفوسفات جاما لحالها.
ممتاز ممتاز ممتاز. بس هم طبعا لما بيتقسموا ما بيبقاش اسمهم بيتا وجاما، هم بيبقوا فوسفات بس وخلاص، صح كده؟
مظبوط.
تمام. ايوه اتفضلي.
شو هو الار ان تي بي؟
رايبونيوكليوتايد، اللي هم الاي والسي والجي واليو. اوكي
دكتور. طيب الريميند الار ان اي اللي بتضل بتصير ار ان اي هي بس 1 فوسفيت؟
طبعا، ما انت شايفه الستراند اوريدي بيبقى فيه 1 فوسفيت اونلي، صح between each two nucleotide.
طيب والريميننج 2 فوسفيت خلص بيروحوا ولا انه بيعملوا كونكت انه خلص راحوا من الار ان اي؟
ايوه راحوا، بس هم بيتم استخدامهم تانيين عشان نبني بيهم الرايبونيوكليوتايد. انتوا عارفين مثلا زي الادينوسين مونوفوسفيت، لما ياخد واحده فوسفيت يبقى ادينوسين داي فوسفيت، ياخد واحده يبقى ادينوسين تراي فوسفيت، صح؟
Yes.
فthis phosphate is recycled to be used again، صح كده؟
Yes.
اوكي.
دكتور هم بصير عندنا ديهايدريشن معاه، حدا بيختار OH حدا بيختار H، بيروح H2O.
Yes. Just hold on please. اوكي يا شباب نكمل. في حد كان بيسأل حاجة قبل ما نكمل؟ اوكي. So let's see here. So in a DNA, when a DNA, you can see this is a structure of a DNA. It's a double strand. You have one single strand from 5 to 3, and the other is other way around. Normally, the gene start with nucleotide donated as +1. As you go downstream of the gene, you have +10, +20, +30. So that's the position of the nucleotide on the DNA. If you go upstream of the gene, a region which is usually called a promoter. So this promoter is a DNA sequence which is upstream of the gene of interest. Usually it have negative sign. So any DNA sequence that have a negative negative sign in the sequence, so you understand that this DNA sequence is before the start of the gene. And you mark the start of the gene with +1, okay? So RNA polymerase needs a promoter sequence to set and localize the gene. And as we said, the site on the DNA template at which RNA polymerase begins the transcription is +1, because actually this is the start of the gene. So downstream denotes the direction of the template. So actually the RNA will be transcribed from that template. Okay? يبقى هي بتبلغ، عشان برضه نوضح الحته دي. الار ان اي بوليمريز بيقعد على منطقه البروموتر، لكن بتبلش من +1 والماسنجر ار ان اي بيطلع من +1 وطالع لغايه نهايه الجين. اوكي؟ طيب. Upstream, of course, will denote the opposite direction because it's actually the region of the promoter sequence. So the nucleotide position in DNA sequence downstream from the start site, which is +1, are indicated by a positive sign, as we said, while the upstream are denoted by a negative sign. Because of course, as you know, as we just said, that the RNA is centralized in a 5', 3' direction, so the RNA polymerase moves down the template of the DNA in the direction of 3', 5'. So actually, the RNA actually moves with respect to the template on the direction of 3', 5'. But the RNA is synthesized in the direction of 5', 3' itself. So as you can see, we have two strands. One is template and one is non-template. And of course, the template will be transcribed. So usually the RNA polymerase will sit, for example, here and will move this way, for example. So actually it moves this way. So the RNA polymerase is formed from 5' to 3'. While the RNA polymerase itself, moves on the template, and the template direction is the opposite, 3', 5'. Okay, شباب؟ في حد عنده سؤال؟
Okay.
Yes.
يعني التمبليت ستراند هو الكودينج سيكونس صح؟
طبعا هو كودينج طبعا. كودينج لو هو هيطلع ماسنجر ار ان اي يطلع منه بروتين، لكن في بعض الدي ان اي سيكونس بتطلع ار ان اي ما بيحصلوش ترانزليشن، يبقى نون كودينج. فهمتي؟
Yes, yes.
Okay. So let's talk now about the transcription.
Promoter is non-coded صح؟
نعم.
Region.
نعم.
The promoter is a non-coding region.
Of course, non-coding. Correct.
Okay.
Let's talk now about how transcription occur, and we'll start with the prokaryotes. Generally, you have three phases: initiation, elongation, and termination. We'll start for the prokaryotic initiation. So you have a DNA sequence here. You have an RNA polymerase. Green circle represents the start of the gene. Red circle represents the end of the gene on the template. So you have an RNA polymerase, and of course you can see that the RNA polymerase sits on the promoter region, which is the DNA sequence that is upstream of the gene. And this RNA polymerase needs initiation factors, IF, initiation factor, to recognize this promoter sequence. And in this case, the complex between the RNA polymerase and the DNA is called closed complex. Why it's called the closed complex? Because the DNA is still closed, it's not melted yet. So you have an upstream and you have a downstream, and the green represents the +1, the start of the gene. Now, the RNA polymerase have the capacity in prokaryotes to melt the DNA, melt the initial sequence, and will form, you can see here, it will form like a bubble, transcription bubble, which is around 12 to 14 base pair. Okay, so in this case, the complex now, it's called open complex. Why it's open complex? Because DNA now it's melted. The initiation is complete when the first two nucleotides is being transcribed from the template. Once the first initial two initial nucleotide is transcribed, now the initiation phase of prokaryotic transcription is terminated. After initiation, we have elongation. Now the polymerase starts to move. How did we know that it moved? Because we can see that the nascent RNA now have been emerged outside, and now it's moving away from the green, which represented the start of the gene, towards the red. During this elongation phase, the transcription bubble or the melted region is now from 14 to 20 base pair or nucleotide, this melted region. Originally, it was 12 to 20. And the RNA polymerase moves one at a time, moves one nucleotide at a time. And you will have a region in which the RNA, you can see the red RNA binds to the blue template. This region represents the DNA RNA hybrid region, and it represents 8 nucleotides. Okay. Once reaching the end of the gene, the termination phase finishes, and this at that time all the complex is released, and now the RNA, the completed RNA strand is released in the nucleus, and the RNA polymerase is recycled back to do another transcription route of the same gene or a different gene. في حد عنده أي سؤال يا جماعة عن الجزء ده؟
Yes, doctor.
تفضل.
بس 14, 20 and 8 are these the length?
The 14 and 20 base pair, it is the length of the melted region, this one. While 8 nucleotides, it is the number of nucleotides that form the double strand between the RNA and DNA. Okay?
Okay, thank you so much.
You're welcome. اوكي نكمل.
دكتور الار ان اي بس.
نعم. نعم.
Doctor, what do you mean by one nucleotide at a time?
مش ال RNA polymerase بيعمل transcribe based on the complementary to the DNA.
Yes.
هو ماشي على الجين ينفع يحط 3 نيوكليوتايد مره واحده؟
حط واحده
واحده بالظبط. نعم. صوت واطي مش سامع خالص. علي صوتك مش سامع السؤال. في سؤال؟
يمين كود ترانسليت لكن دي اصلا non coding الاثنين هو الفرق في الناتج العلمي خالص. الناتج العلمي طالع يحمل الاشاره لخلطه بروتين يتخلل تنفيذ كان ناتج علمي في الخاص من السايت ناتج علمي في الخاص Transcription هو عملية التسليم translation only the structure of messenger RNA is shown in the derivative transcription sequence, then the messenger RNA is the genes are scattered nuclei, but the genes are all in one in the sequence. So you have one single RNA that is transmitted to 5 proteins. Here in eukaryotic you have 5 messenger RNA that is transmitted to 5 proteins. But in the material, almost the same. Finally, eukaryotic is used or recognized in operate. Okay? You apply genes بتاعت المثال بتاع التراكتيفين. Every gene has a different name. Other genes have a different name. Okay? Let's say the flexible. A, B, C, D. It's only a 1, 2, 3, 4, 5. Okay? Have you ever heard of it? هو المين جدا كتير total one promoted. So if you have 2 another difference when you go back. You have more chemical connectivity. Important difference. They have always relation, but it's single material and different types of genes. They have always relation, it's single material and different types of material. While the only relation single material one and different types of functional elements, lysosomal elements, are other small components. So in this case, you can see the difference between the phenotype of the RNA that is nucleotide, the RNA that is transcribed, validation of these RNA, and obtain the RNA that is. So now we talk about the transcription gene nucleotide. Transcription nucleotide. So we have next to do and actually this actually the genetic of the gene of the eukaryotic gene. In transcription, you need something called transcription factor. In particular, initially, the transcription activity transcription activity assume that in the nucleus itself we have RNA relate 1, RNA relate 2, RNA relate 3. So we are dealing now with transcription that is using RNA relate 2 to produce messenger RNA. So any transcription factor RNA relate 2 is the name 2 here, that's like 2. So that's transcription factor B. So this transcription factor B consists of the feature. The factor that binds protein, which is the PGP, the glucose, and the protein factor that binds protein. So all of these factors is called transcription factor B. And the binds of the factor, what is this factor? Factor is actually the promoter and the sequence is very rich in so the first step in eukaryotic transcription is binding the CSC to the data the promoter region of the cell. The CPP, which is the part of the data box, it is the first protein to bind to the data box. And actually the center of used to be in the in the gene. This is the binding of the gene. While the second box of the transcription factor has constantly initiating the transcription from promoting the log. So you can think that there are some chemicals in the cell that don't have the factor box. So why do you have the start? Why do you have the start? The transcription factor is the first to make the once the TSC bound to the data box, another two transcription factors, TSCA, TSCB, you can see the stop more than the factor, and you will form the subject which is called upstream genetic syndrome. Why is it called upstream genetic syndrome? Because you can see that the blood line here and the gene is deep. Actually, the subject is sitting before the part of the gene. That's why it's called upstream genetic syndrome. The cellular upstream that consists of the upstream genetic syndrome actually consists of three transcription factors. A, C, and the original C, bound to the gene. Actually, the TSCB binds to the major group of the gene and will help the polymerase in mounting the DNA strand of the transcription factor. While the transcription factor 2S, another transcription factor, is bound with the unable gene and will help to keep structure in the recruit the unable gene to be something. And now you can see that the unable gene, the W transcription factor 2S, will keep on the gene and you will form something called 4-PIC. And what 4-PIC? PIC is called pre-inclusion function. Now the technique is formed of 1 minute series, what common transcription factors 4 transcription factors. Transcription factor 3, 1, transcription factor 2S, 2, transcription factor 2B, 2, transcription factor 2S, 4. So now you have in the subject, in the 4-PIC, primary two, but general transcription factors are 4, bound to the promoter region, upstream of the gene, and is ready now to receive transcription, and this complex is called 4-PIC. Okay. ready for transcription. صح ممتاز. يفضل حاجه واحده بس جدا معلومات مهمه برضه ان ده البايندنج بيحصل من البايند من خلال الميزه. الحاجه الثانيه كمان اللي الترانسكريبشن فاكتور سي اس هو اكشلي اللي ريكروت الار ان اي already fixed. So we need to know the core DIC concept. Ready for transcription. This after that, another transcription factor is transcription factor 2. The second cell in the cell TFC. TFC is one of the very important transcriptions. So TFC recruits TFC. This differentiation needs to divide it. And now the complex is called flow. And why flow? Again, because the DNA is not nice. So we have additional two complexes. Now the DIC, the DNA second complex, first is assembled at every subtype of every protein coding. So that means that these complex cannot be assembled at even that will not be coded, that will not be protein. After that, we have the FTS has two very important steps. The timing property and the helical step. And the first, the helical will help in making the disease. And you can see here the transcription bubble starts to appear. That's why now the complex is called open CIC, open field system. And why it's called the field system complex? It didn't work yet. And as well as the transcription factor TX we have, the kinase that means it doesn't factor control. This is no problem. Surrounding the first site now is melted. As I was and the template now is open to transcription. And you can see here that the RNA as the polymer transcribed and moved away from the promoter, the transcription factor protein is released from the RNA. And you can see here that the TFCS, the one with the kinase activity, encapsulates the terminal end of the RNA release with construction terminal domain of the RNA release. And this phosphorylation of the terminal end of the RNA release will provide lots of elements needed by this method to move over the DNA to do the transcription. Once the template starts to move, it will enter the elongation phase. That's why the initiation factor the transcription factor are used, and another set of elongation factors are used by the complex. Now, polymerase will initiate transcription of this cell, and all the complex will move away from the promoter to start transcription of the cell. So let's المثال ببساطة احنا عندنا transcription factor أولاني اسمه التي هو اللي بيسحب التي اس عشان ينزلها على ايه اهميه التي اس؟ التي اس ده بيساعد في تحسين جدا ممكن تبقى تحول التي اي سي يبقى فلويد ولا اسمه فلويد all in level service يساوي الضغط عند الفلويد بالتفاصيل بتاع transcription of translation into flow. طيب الريليتيف اكتيفيتي الاس اس الاساسيه من الريليتيف ديفينيشن اكتيفيتي او التسويق اكتيفيتي ديفينيشن كل مثال في اس اس ده بتاع تكنيك لازم يجري على كده ان البابل ترانسكريبشن بابل الثاني يتحول من سي اي سي الى اوتو سي اي عايز تثبت almost to move over the DNA structure. انت هنا اللي حصل فيه بتاعت عمليه الموت ان الترانسكريبشن بتاعت السي اي انت اللي بتكتب يجي للجنين او منطقه موجوده تحت الارض اللي اسمها سي اي سي اربط في الجنين في دي structure related التركيب الثلاث structure الجنين structure related دي structure related كثير بيتغير في structure related ولما يتحول السبجكت من اوبن جي اي سي انتو كونكتوري هذا يجيب الكونكتوري implementation factor most important. So that's why you see that the most implementation factors are نستطيع هنا بس بمرحلة الإي سي يعني الموضوع بعد كده معقد أكتر تعقيد كبير جدا في الليفل بتاعه assembly of the subject. And every transcription factor there is a concept of it. This figure actually shows the green arrow shows the green with green. While all these structures, this is the conceptualized region. Every single line is used to one material. وعايز اقول لك الكلام الكتيره دي ان كل واحد ما بيختلفش عن تفكير مره واحده هو بيختلف عن تفكير اكثر من مره يعني هو تعمل اي شيء في الريجن وانت قارن مثل ما يمكن خلي يفكر يا جماعه مثلا حاجه زي مثلا لك كده يعني الانسولين بيفتح الكلى او كل شيء خلي الانسولين مره واحده هيقدر يفتح فهي الجسم اللعب كده ده كله كان بيستخدمه ازاي ممكن يعملوا ياخد برضه نفس الطريقه عن نفس الامتحان بس قال في بكتيريا person only needs to memorize. And usually the key functions, all these functions, in the sequence of the key functions. The key functions, the functions, are we find that the element of changes equalize is very low at 1 to 55%. In this figure, we see how we find all these unparameterized طيب حد عندنا تحت هنا يقول لي في درجات الكلمه ولا لا؟ اتفضل. مين اللي بيساعدك تعمل الهرمون في ولا حاجه. اخر نقطه بيقول لك موبايل جديد فاهم تبقى المحاضره الجايه بنتكلم على الكلمتين فاهم. Now we go to the beginning of the slide to talk about the يعمل عايزين السوبر غني برايمري. ما بقدر اقول عليه دلوقتي ممكن يبقى سؤال هو ليه ربنا خلينا نكمل الفكره دي ممكن ارجع لك فيها بس هو الفكره دي Rna is that Rna is the structure that protects the 5 points of messenger RNA. Did you find that difficult? Rna is that the 5 dendritic structure that protects the 5 points of messenger RNA. While the polyethylene will protect the polyethylene will protect the 3 points. Then there are slicing which remove the input and only leave functional coding. كل اللي بتشوفه هنا كله بالتفصيل. بنجيب لك ولا بتحاول لازم تحط قبل ما يطلع قبل المايجريشن كان بالنسبه ممكن ان الار ان اي لازم يطلع من النيوكليس هو دي multiple related proteins from a single gene by means of the connection. يعني في صفة هنا إن ربنا بيستخدمها ultimately يطلع multiple proteins from the same protein from the same يفهم الكلمة different cell types. الناس اللي احنا بنتكلم عليه دي وضح لنا الكلام مثلا بروتين مكتوب اسمه فايبرين. This is the multigen extracellular adhesive extracellular adhesive over the line and can be secreted by cytoplasm and the deposit. And it is a very good example of the genetic. However, protein characterized by the cell translation to cytoplasm. But in both cells, although it comes from the same gene, both cells but it behaves different in its height. These cytoplasms, the cytoplasm contain two exons, CCA, CC, and these two exons will encode a protein that enables the cytoplasm to adhere. So these two exons will give a sticky behavior to the cytoplasm, which is secreted by the cytoplasm, to help the cytoplasm to stick to the cytoplasm. While in the hepatocyte, the EC and CCG exons of the same gene, the hydrogen, will behave as a being entry. So the cytoplasm, which is secreted by the hepatocyte into blood, will not adhere to other cell types. النقطة دي كويس على الكتاب. inside this block, you can see this active messenger RNA that is transcribed from these two in fibers. And you can see the functional messenger RNA in the fiber block will contain E3D and E3A. While the messenger RNA in the hepatocyte, it doesn't contain E3D and E3A. يعني إن E3D لما اتكلمت عن تكتشف المسجد تم معاملاتهم في طريقة بتاعت الصوت على أساس إن هم وتم ربطهم على الإسلام، لكن في طريقة الصوت الصوت تم معاملاتهم على إنهم جدا لأن لهم فانكشن يخلوا هذا البروتين هذا الشكل جديد. فأنت تفضل يا لاحظ إن دول خليجين مختلفين. يبقى في الفايبر بلوك يبقى الفايبر ممكن هذا عنده الشكل بيهيفير. أما في الهيباتوسايت الفايبر ممكن بيجيب في consistency. It's not organ but it's behavior due to the lacking ECG or ECA. تمام؟ هي مالك خلاص؟ طبعا العربيين لما يتكلموا ترجمة بتاع زي الفايبر الفايبر كلاس هيجيلك بروتين وشوية ECG while the message are used when it's quantified in the hepatocyte it will form a fiber mission that links the behavior of the fiber. إنما زي ما جماعة فهمت ما بتوضحوا إيه الفايبر وكده الفلتر في حد عنده سؤال؟ Any more questions? Keep the practice. Thank you very much.